gentlemen. I'm Ed Byrne, the President and Principal at King's College London, and it's a huge pleasure to welcome you all here this early evening for the 4th annual Higgs Lecture, which will take place shortly. The Higgs Lecture is the most prestigious lecture within the Faculty of Natural and Mathematical Sciences, and each year we focus on a different area of research in the faculty, which of course includes chemistry, informatics, mathematics and physics. The inaugural lecture was in the area of physics, delivered by our eminent alumnus, Professor Peter Higgs, who won the 2013 Nobel Prize in Physics for predicting the existence of the Higgs boson, and kindly agreed to give his name to this lecture series and also to launch it. Last year, we were joined by Professor Caroline Series, an eminent mathematician who delivered a lecture on hyperbolic geometry. This year will be the first lecture in the area of chemistry, and I'm absolutely delighted that Professor James Bowie at the University of California, Los Angeles, UCLA, has agreed to join us this afternoon. Welcome, Professor. Professor Bowie has had a distinguished career and is an internationally acclaimed chemist and biochemist. Please let me now hand over to Professor Paula Booth, head of our Department of Chemistry, who will formally introduce this evening's lecturer. Paula.
Hello and welcome, everybody. So I'm absolutely delighted to introduce Professor Jim Bowie as our Higgs lecturer this year. Before I carry on, though, I've been asked to point out the fire exits. Now, that's not for a rapid departure as soon as Jim stands up here. That's if we really think there's a fire and we need to leave and save our lives rather than listen to what he has to deliver. So Jim is Professor of Chemistry and Biochemistry and Vice Chair of his department at the University of California, Los Angeles, or UCLA, as we all say. He did his undergraduate degree in chemistry at Carleton College, which I believe is in Minnesota, which is where he's from. He did his PhD at MIT, Massachusetts Institute of Technology, with Bob Sauer, and that's when he started his work on protein structure. He then moved to UCLA as a postdoc with David Eisenberg to do more work understanding and predicting protein structures. And following that, he started his own independent career at UCLA in 1993, and I'm guessing he's been there ever since, because there's nothing else to tell me he's been elsewhere, not even back to Minnesota, maybe for the odd ice hockey game. He's won many awards. He's been elected Fellow of the American Association for the Advancement of Science in 2008. He's given many distinguished lectures. He's also done great service to the community. He's organised lots of meetings. He's been the chair of a number of committees, both nationally and internationally, one of the more recent ones being president of the Protein Society. His research still focuses on proteins, understanding how they adopt their specific structures for function, how they interact with each other, and how they can be harnessed to produce biofuels. And he's here to talk about his work on membrane proteins, and he's been a real pioneer in this area, one of the few people in the world doing this kind of work. These proteins account for about a third of all the proteins in the cell, and they dominate drug targets. Jim's work beautifully combines chemical methods with biology, and today he's going to tell us about some of his groundbreaking work to investigate the mechanisms of how these proteins achieve their functional structures. So, Professor Bowie, it's with great pleasure I invite you to present the 4th annual Higgs Lecture toward quantitative folding studies of complex membrane proteins. And I hope we don't get the feedback.
I'm a little nervous about turning this on. So far, so good. It's on. We're okay, and no feedback. Excellent. I have to say it's just an amazing honor to be invited to give this lecture here at this historic university, and it's always a pleasure to have an excuse to come back to England. I actually have a lot of roots here, actually. My father is English, grew up here, and two of my siblings were actually born here in London. My father was a great storyteller, and so many of the stories he told were of England, and it's always great to sort of see, be in the environment where those stories took place. Not to mention the fact that this is really an amazing country. So we've had a really great time visiting again. So thank you for that. All right, so what I want to do today is tell you about our efforts to understand how a particularly important and interesting class of proteins fold. And I know that there's a lot of highly sophisticated structural biologists in the audience, but I've also been told that it's a fairly general audience, and I know that's true as well. So what I'm going to try to do today is I'm going to try to tell you, explain in the beginning anyway, why protein folding is amazing and why it's important before I dive into the specifics of what we do. And I've tried to intersperse a little bit of background, more background than usual. So I hope the two camps here will bear with me a bit. All right, so let's go back to 1958 when this rather famous Englishman, Francis Crick, you might have heard of, proposed the central dogma of biology in which information flows from DNA to RNA, and this RNA encodes a string of amino acids in a particular order. And these amino acids have different properties. Some are polar, some are charged. And this order of amino acids has the amazing property that for many of these proteins, they can actually adopt a particular structure. And they adopt many different beautiful structures. I find them really fun to look at. And these structures basically place atoms at particular places in space that allow these proteins to interact specifically with other molecules and catalyze chemical reactions, or just bind to other molecules and create structures in the cell. So they're responsible for carrying out the instructions of the genome. They're the worker bees of the genome. Okay, now the fact that they can adopt these structures is really quite remarkable. So let me just try to explain why I say that. So this is the first protein structure determined of a protein that can catalyze a reaction, an enzyme. And it was solved right here in London in 1965. So this is a fairly small protein. It's about 126 residues, amino acids, but it's 1,000 atoms. It's 1,000 atoms, and they're all placed in a unique place in space. That's pretty incredible, okay? Look at this boy. This guy is gigantic. 200,000 atoms, and they're all 200,000 atoms all placed in space in the right place. That's incredible. We want to understand how that happens. Here's our lysozyme right here, this first structure that I just showed you. Okay, that's pretty incredible. So how does this happen? Well, there was a famous experiment done in the early 70s by Christian Anfinsen, which won him a Nobel Prize, and it's an incredibly simple experiment. He took a protein, put it in conditions where it would unfold. It unfolds, and then he just took it and put it back into water, and the protein spontaneously refolded. So what does that mean? It means that the information about the sequence, the information about the structure is entirely encoded within the sequence of amino acids, within the molecule itself. Okay? So that means it is basically a physics problem, and it's something you can study in a test tube, and so that's really launched the protein folding field. Okay, so, but it's a very hard physics problem. So let me try to explain why that is. So, so proteins adopt a particular conformation, but these are polymers that have a lot of flexible bonds, and so there's a huge number of possible conformations that they could adopt. Gazillion is American for a lot. So there's a lot of ways it could fold up, but it finds one. So in other words, there's a huge entropy cost to folding a protein. And it's estimated in terms of free energy that this, for a 100 residue protein, something the size of that lysozyme molecule, it's on the order of 100 kcals per mole just to fix the chain in one conformation. Okay, so in order to basically stabilize this structure over all the other possible structures, you need to get 100 kcals per mole of stabilizing interactions in this state compared to all the other states. And that's a lot. So how does that happen? Well, these amino acids all have different properties. Their side chains, some are apolar, some are charged, some are polar. And when the protein folds, the apolars tend to go inside the protein, and so you get favorable release of water, the hydrophobic effect. You also get nice van der Waals packing interactions in the core. You get salt bridges and hydrogen bonding interactions in the folded state. Now, all of these are non-covalent interactions. They're fairly modest in free energy, on the order of a kcal or so. And so in order to stabilize this state over all the other possibilities, you need to have a lot of these interactions. And proteins can do that because they have this variety of amino acids in a particular order that allows them to make all the interactions it needs to make. But that's hard. Biological polymers are really unique polymers because they can adopt a particular structure. Most polymers can't do that. Okay. Now, the other amazing part about this is that it's a really hard search problem. Again, there's a huge number of conformations over here, and somehow the chain has to find this one structure. And Cyrus Leventhal did a really simple back-of-the-envelope calculation to sort of illustrate the huge number of possible conformations that are possible. So what he said was, let's imagine we could sample 10 to the 15th conformations per second. That's ridiculously fast. That's faster than a bond, order of magnitude faster than a bond vibration. So that's can't be done. But even if you could do it that fast, it would still take many orders of magnitude longer than the age of the universe in order to sample all possible conformations, believe it or not. So proteins fold on the order of microseconds to seconds, so they clearly can't sample all of conformation space. So there must be some sort of order or some sort of pathway that allows them to find that state, that conformation in the sea of possibilities. Okay? And that's something that many people have been trying to understand over the years. Okay, so it's a pretty amazing thing that proteins can fold. And they do a great job of it. It's also important. Okay, why is it important? Well, first off, it's a fundamental biological process, obviously, and if we want to understand biology, we need to understand everything about it. Okay, so it's a major process. Okay, the other thing is determining these structures tends to be difficult experimentally, and some structures we really can't get to, large flexible structures. So it'd be really great if we could learn how to predict accurately a structure from sequence information, because it's really easy generally to determine sequences, especially these days. So there's a large effort to try to understand folding so that we can predict structure. We also want to understand how evolution occurs, and one of the major constraints on protein evolution is just simply maintaining their structure. Right? That's a major constraint on the types of amino acids that you can put in there. We want to be able to design proteins to do what we like them to do. So the cells have produced all these proteins to do all these complex reactions. We'd like to maybe have them do things that we like them to do, maybe do a chiral synthesis. We'd like to build nanobots and things. We'd like to build arrays of molecules, things like that. In order to do that, we need to, part of that process is making sure they fold properly. Okay, so part of design is also folding. And then finally, most genetic mutations that affect protein sequences affect that cause disease affect the ability of the protein to fold, rather than directly affect the function of the protein. So if we want to intervene in those diseases with drug design, it's important that we sort of understand clearly exactly what's going on, what's the folding defect, so that we can try to fix it. Okay, so as a result, it's not only amazing, it's an important process, and so as a result, there's been an enormous effort over the years to understand how proteins fold. Hundreds of labs over the last 50 years, okay? But 99.9% of that effort, literally, has been directed at proteins that fold in water. So proteins in the cytoplasm, proteins that are out in the extracellular space that are buzzing around in our bloodstreams. And and that's great, but there's a lot of other proteins that float around in membranes. So cells have a lot of membranes. So it's plasma membrane, there's lots of organelles in there, and in these membranes are lots of proteins that float around in there. And these proteins play very important biological roles. They're important for communicating with the outside world and telling the cell what to do as a result. They're important for bringing molecules inside and outside of the cell, for communicating between organelles and bringing molecules between organelles. They play really important biological roles. And about 30% of all proteins are membrane proteins. Okay, so this is a huge class of proteins that is playing very important roles. They also have a folding problem, but it's been largely ignored. And it's been ignored because these are a real pain to work on. Nevertheless, if the folding problem is important, it's also important for these proteins, and we've got to make progress on these as well. So that's what we've been trying to do. All right, now in many ways, the membrane protein folding problem is much more complex than the soluble protein folding problem. Soluble proteins fold in a homogeneous environment, water, which is complicated enough, but it's much more complicated for membrane proteins. So in the center of the bilayer here, it's apolar. On the edge of the bilayer, near the head groups, it's polar and charged. And then they also fold in bulk water. So this makes things complex from a theoretical perspective because the driving forces for folding are going to be different throughout the different parts of the bilayer. It also makes it difficult to makes it difficult to work with them experimentally because we have to keep different regions of the protein happy, and they have different folding forces. Okay. So the hydrophobic organization of membrane proteins is very different than soluble proteins, so I just want to illustrate that. So this is a helical bundle, a soluble protein helix bundle. And what I've done is I've colored the hydrophobic residues in green and the hydrophilic residues in red. Okay, so now if you tip this down and look down its axis, you see that the hydrophobics are pointing inside the protein. So you get this apolar core of the protein, and then the surface of the protein is polar. Okay, so basically you're shielding the apolar stuff inside, and the protein surface is polar and can float around in water. Now, membrane proteins are much more hydrophobic. So this is a membrane protein, and you can see it's quite greasy. If you tip it down, you can see that the outside is very apolar. The center of the protein is somewhat more polar, but it's still quite greasy. Okay, now when we work with these proteins, it's inconvenient, I would say, to work with them in membranes. Okay, although I'm going to tell you how we're working with them in membranes now. But in any case, it's difficult to do physical studies of membrane proteins in membranes. So typically, what we do is we solubilize them in detergent. So we add molecules that look like this. They have a polar head group and an apolar tail, and we add that to the protein. It dissolves the membranes and the protein, and basically coats the protein in a polar shell. So the detergent organizes around the apolar part of the membrane, and then this detergent protein complex can now float around in water, and we can do physical studies on that. Okay, so the apolar bits are shielded by this detergent, and then the water-soluble bits can interact with water as they did before. Okay, so this makes them convenient to work with in a sense, but it also is a problem because a lot of membrane, these are not membranes, and a lot of membrane proteins don't like that. That tends to make them unstable, and they frequently aggregate. So it's sort of an expedient. It's not ideal, but it's something it's something that we do quite frequently. And again, I'm going to tell you how we're starting to work with membrane proteins in membranes towards the end. Okay, so these are very hydrophobic sequences, and so they're generally not made in the cytoplasm and then inserted into the bilayer. They're generally inserted co-translationally. Okay, so here's a ribosome making a polypeptide, and there's a conversation between this membrane protein, the translocon, and this nascent polypeptide that decide what parts of the protein get inserted into the bilayer. And this is largely based on the hydrophobicity of the sequence, how greasy it is. Once that protein is inserted into the bilayer, it can complete folding to its final folded form. And so this is the part of this process that we want to understand. How does it get to this final folded form? And this is going to involve complex interactions within the protein itself, and also between the protein and the bilayer that we'd like to figure out. Okay. All right. Now to study folding, this second stage of folding, it's essential that we have reversible conditions, or to study folding in a quantitative way. It's really essential that we have reversible conditions where it can go back and forth between the folded state and the unfolded state, so that we can measure things like thermodynamic parameters and kinetic parameters, so that we can learn about kinetics, learn about something about the mechanism of folding. Okay, and that's just really hard to achieve for membrane proteins. And another problem for proteins in general is that if you're a self-respecting protein, you're going to be mostly folded. Okay? And so there's going to be a tiny amount of this unfolded state around. And so it's very hard to study this folding process when you've only got this small fraction unfolded. So generally what we try to do is we try to find ways to push the equilibrium in favor of the unfolded state so that we can increase its concentration so we can study this. And so we add denaturants. Okay, so typical denaturants are urea, guanidine, or temperature. Okay, so at low denaturant, the protein is completely folded, but as you go to higher and higher denaturants, you can drive it to the unfolded state. And it's in this middle region where we can measure things like thermodynamic parameters and start to look at kinetics. To look at kinetics, you can jump to high denaturant, watch the protein unfold over time, and if you want to look at refolding, you can start at high denaturant and watch the protein refold over time. Okay, so these kinds of techniques are just very commonplace for soluble proteins. Hundreds of soluble proteins have been looked at in this way. Okay? But it's just really hard to get conditions where you can do this for membrane proteins. And there's very few examples of that. Paul has been a pioneer in trying to develop techniques for other model systems. But right now, or for the last decade or so, the best model system by far is this protein bacteriorhodopsin. We have a crystal structure, we can get reversible folding, and we can make mutants of the protein. So we can do a lot of things that are really commonplace now for soluble proteins with this particular membrane protein. So this has sort of been a workhorse protein for us and other labs over the years. It has this nice bound retinal chromophore that we can use as a monitor for folding. So so the denaturant we use for this protein is a denaturing detergent, SDS. So rather than use urea or guanidine as you would for a soluble protein, we drive unfolding with a detergent that denatures the protein, SDS. So this is just plotting this retinal absorbance as a function of SDS concentration, and we get these very nice unfolding curves. So this is the wild type in black. Here's a stable mutant of the protein in blue, and an unstable mutant of the protein in red. Okay, so in these transition zones here, we've increased the concentration of unfolded enough that we can actually measure equilibrium constants. And so we can compare those equilibrium constants for folding with the wild type and the mutants. Okay, so using this approach, then we can actually measure the thermodynamic contribution of side chains to the stability of the protein. So that's been very useful to us over the years. We can also start at low SDS concentration and jump to high SDS, watch the protein unfold, and go back and watch the protein refold. And the pioneer in these kinetic experiments is right here, Paula Booth. She's all embarrassed now. So she's done a lot of classic work on the folding of bacteriorhodopsin. In fact, I will credit her with really starting the modern age of membrane protein folding, because she's the first one to really look at kinetics of folding of a membrane protein, trying to understand the pathway for folding. So we've been following in her footsteps. Okay, so when we think about protein folding, we have a number of basic questions in mind. We'd like to know what the native state looks like. We'd like to know what the unfolded state looks like. we'd like to know what stabilizes the native state over the unfolded state. And we'd also like to know something about the pathway. In particular, can we learn something about what the structure of the transition state? Okay, now with bacteriodopsin, because we have all these nice tools for this particular protein, we've been able to address most of those questions. So my lab, and Paula's lab, and other groups have addressed most of these questions. So we've learned the high-resolution structure of the native state through distance measurements using EPR measurements in the unfolded state. We've learned a lot about what the unfolded state looks like. Paul has pioneered using mutants, the effect of mutants on kinetics of folding and unfolding to learn about the structure of the transition state. And then we've done quite a bit of work trying to understand what stabilizes the native state over the unfolded state. And I thought I would just tell you a little bit about our work in this area, some of the highlights. Just to give you sort of a sense of the kinds of things we can do with this particular system. Okay, so the first story I'll tell you very briefly is an issue of how strong are hydrogen bonds in membrane proteins. And this is the work of a former graduate student, Nathan Joe. All right, so when we started this work, the dogma in the field was that hydrogen bonds in membrane proteins should be very strong, much stronger than they are in water-soluble proteins. Why is that? Well, if this is a hydrogen bond in a protein, if you break that hydrogen bond, you can easily form hydrogen bonds to water. Okay, and so these hydrogen bonds to water basically compete for that hydrogen bond. So that lowers the net contribution of that hydrogen bond to the stability. Okay? Now, if you put this in a membrane, you don't have much water around, and so you don't have that competition for hydrogen bonds, and so you should get the full effect of that hydrogen bond. Okay, so we thought that these hydrogen bonds should be very strong, and people estimated that these could be worth on the order of 4 to 5 kcals per mole. So very strong interactions. Now, if you look in membrane proteins, you see these networks of hydrogen bonds across these helices. So I've just shown the number of hydrogen bonds across each of these helix pairs. So there's a lot of these hydrogen bonding interactions. If these are worth 4 or 5 kcals per mole, that's getting us pretty close to that 100 kcals per mole that we need. So this could be really a dominant driver of membrane protein folding. Okay, so that's really what we thought was true, but we were now in a position we could actually test that hypothesis. And so we did that. We basically made mutations in all these hydrogen bonding interactions, and I can't tell you how we did this. It was a little more complicated than just making mutations. But in any case, we measured interaction-free energies between all these hydrogen bonding interactions in bacteriodopsin. And these are the kind of numbers that we got that are shown in these boxes. Okay, so a couple are reasonably strong, nearly a couple kcals, but most of them are kind of in the noise of the experiment. Okay, they're certainly not the 4 or 5 kcals per mole that we were expecting. Okay, so this led us to scratch our heads a lot, and we realized after worrying a lot that really that picture that I just presented was really quite naive. These hydrogen bonds are not being made in a membrane environment. They're being made in a protein environment. They're surrounded by protein. Okay? And that's much more polar than that apolar membrane environment that we had envisioned. In fact, if you look in the interior of bacteriodopsin, right here, there's tons of water in there. I've just circled all the water molecules that are in there, in this part of the protein. So, if you break one of those hydrogen bonds, you can probably find another water molecule to compete with it. Okay. The other thing is that the protein itself is also polar. Okay, so here's our hydrogen bond again. If we break that hydrogen bond, you can generally find the backbone of the protein is polar, and you can generally find a hydrogen bonding interaction to the backbone. Okay, so in order for that thing to be worth a kcal, it's got to be stronger by a kcal than that hydrogen bond. Okay, so the upshot is that basically there's a lot of potential for competing interactions, polar interactions in the core of the protein that we really didn't think about before. Okay, so we don't think, hydrogen bonds are certainly a net favorable interaction in membrane proteins, but we don't think that these are dominant drivers of membrane protein folding. Okay, so if that's true, then what is? Well, in the membrane, you don't have this hydrophobic effect that you have in water, which is a dominant driver of soluble protein folding. So I think you're sort of left with van der Waals packing. Okay, so if that's true, then you might expect that memory proteins would have more optimized van der Waals packing than soluble proteins, because that's the energy currency that they have to spend, and they're going to try to spend it wisely. Okay, so we decided to investigate that, and again, this is the work of Nathan Joe with Ahmed Oberai, former graduate students in the lab. Okay, and we envisioned two possible ways you could optimize van der Waals packing. One is, so if this is a soluble protein, this is a memory protein, perhaps you pack more densely. Okay, so you have fewer holes and cavities in the center of the protein, so you get more bang for your atom buck. Okay, the other possibility is that you just build a different structure. Basically, you adjust the structure so that you bury more stuff together. So if this is our soluble protein, maybe you have to slide this helix down so you can bury more stuff. Okay, so you get more van der Waals packing. You don't necessarily get more efficient packing, you just get more of it. Okay, so that's what we're thinking. And so which one of these is true, or are both true, or are none true? All right, so one thing you can do is you can just look at structures. And so people did that before us, and basically it was controversial. So these people say, yeah, they're better packed, and these people say, no, they're not better packed. And basically it just depends on what the technique you use to evaluate packing. Okay, so we decided to look at this experimentally. Could we see any difference experimentally? So the experiment we did is illustrated here. So here's a side chain in the core of Bacteriorhodopsin. Okay, and there it is. All right, so what we did is we delete that side chain by making a mutation. So we make a hole in the protein. Then we solve the crystal structure and learn and measure how big that hole is that we left. Okay, and then using our SDS stability assay, we can measure the thermodynamic contribution of that the effect of that mutation on stability. Okay, and we can do that for a lot of positions in the protein. Okay, so these are the results. So this is just plotting decrease in stability as a function of cavity size. All right, so so the slope of this line reflects the van der Waals contribution. How much van der Waals, how much free energy you lose by leaving a hole. Okay, so this is from a water-soluble protein. This is work of Brian Matthews, early 90s. And then this is what we see for Bacteriorhodopsin. Okay, as far as we can tell, there isn't much difference. Okay, so so if memory proteins are better packed than soluble proteins, it's a pretty subtle deal. Okay, what about this issue of packing more stuff together? Well, that seems to be clearer. So this is just plotting the average surface area buried as a function of protein size. Average surface area buried per residue. All right, so the soluble proteins are shown in green, and then the membrane proteins are shown in red. The transmembrane regions of membrane proteins are shown in red. And you can see that they're really a different class. They're really good at burying surface area. Okay, so we do think that this is something that they do. They basically build structures that bury a lot of stuff together. Okay, so this constraint that they need to bury a lot of stuff actually has some practical consequences, which we describe in this paper, but basically the upshot is that when they have to bury a lot of stuff together, okay, so there's a lot of buried side chains, more buried side chains than soluble proteins, and buried side chains are more restricted than surface side chains, right, because they're doing stuff, they're interacting with other side chains. So it's harder to change them than if they're on the surface. Okay, so the fact that you have a lot more buried residues in the protein basically slows the rate of evolution of the transmembrane regions, and also makes them more susceptible to disease-causing mutations, because the chances that you're going to do something bad if it's a buried residue are much higher than if it's on the surface. Okay, so back to this picture. So we've been able to learn stuff, learn things about membrane protein folding with this system, and some of it has practical consequences. So that's nice, okay, but this is a bit frustrating because for two reasons. One, this is in detergent solution, right? Membrane proteins fold in membranes. And membranes are going to have a profound influence on the unfolded state of the protein and also the stability of the folded state. And so we really need to be able to study folding in membranes if we really want to learn about biological membrane protein folding. And the other thing is this is bacteriorhodopsin. Okay, so this is one protein. It's a fine protein, but it's a very well-behaved membrane protein. Most membrane proteins don't behave as well as bacteriorhodopsin. And the chances that you could take a random membrane protein and do this kind of work on it is, at this point, is essentially zero. Okay, so we really need to be able to move into bilayers. We need to be able to develop techniques where we can expand the range of proteins that we can look at. Okay, so we're trying to do that, and we really want to be able to study proteins like this. So these are a couple of proteins where we know that mutations affect the folding of these proteins, but we really can't, and they cause disease, but we really can't study the folding of these proteins in a quantitative way. We really want to be able to do that under natural conditions. Okay, so we're trying to move that direction, so let me tell you what we're trying to do. Okay, so first let's get into bilayers. So we've developed an approach which we call steric trapping. So what we want to do is we want to drive unfolding in bilayers without really altering the bilayer if possible. So what we do is we label the protein at two places that are close in space with biotin tags. And then we can add a large protein that binds to those tags, namely monovalent streptavidin. So monovalent streptavidin binds to one of those tags with high affinity. Now, if we set this up right, though, a second streptavidin can't bind due to steric overlap. Okay, so the only way the second one binds is if the protein is unfolded. Okay, so the first binding event is high affinity, the second binding event is reduced affinity, and that reduced affinity reflects the delta G of unfolding. Okay, so simply by measuring binding curves then of streptavidin, we can extract unfolding free energies. The other thing we can do is we can jump to high streptavidin concentration and watch the protein unfold. And we're developing an approach now where we can cut these tags off and watch the protein refold. So all the kinds of things that we want to be able to do to study folding, but now in bilayers. So we've developed this technique over the years with increasing degrees of difficulty, and now we're into larger membrane proteins. And this is just an example of recent stuff, still unpublished, where we're looking at unfolding of bacteriorhodopsin in actual bilayers, in vesicles, in these vesicles. Okay, so this is just a binding curve, and from this binding curve we can extract a delta G of unfolding for bacteriorhodopsin. And that's actually the first time the free energy of a membrane protein has been measured in actual bilayers, as far as I know. It's pretty pathetic, but that's the stage of the field. All right. So this is nice. So now we can actually study how bilayer properties affect the stability of the protein, protein like bacteriorhodopsin. But we're still with bacteriorhodopsin. What about those other membrane proteins, those that are harder to work with? So how are we going to get there? Well, a major problem with membrane protein folding is that when they unfold, they tend to be very sticky and they aggregate very easily. And that seems to be a major problem with getting reversible folding. So if we could go to infinite dilution, we could avoid that problem. Okay? So that's what we're trying to do. So basically what we want to do is we want to have a single protein in a vesicle. Okay? So that now when this protein unfolds, it doesn't have any friends to get in trouble with. There's nothing to aggregate with. So we hope that will allow us give us a better chance to see reversible folding for other membrane proteins. And so to do that, we have to move to single molecule fluorescence techniques. So we're basically trying to develop single molecule fluorescence techniques to do steric trapping of single proteins in vesicles. And this is work that we're doing in collaboration with Shimon Weiss, who's a pioneer really in single molecule fluorescence. And Rob is just suffering in technical hell right now, trying to get this to work. But I think you can imagine this is something that should be doable. Okay, so now I just want to spend the rest of the talk, though, on a technique I'm particularly excited about, and that's using forced unfolding, single molecule forced unfolding experiments. And this is work we have done with a wonderful collaborator in Korea, Tae-yoon Yoon. We talked at a number of meetings about this and finally decided to actually do it. And he recruited his wonderful graduate student, Do-young Min, who is now a postdoc in my lab and set up tweezers in my lab. And I managed to convince Rob to help out on this project as well. Okay, so here's the setup. So we have a membrane protein in here, and we attach one end of the membrane protein to a DNA tether that's now attached to a solid support, glass surface. The other end of the membrane protein we attach to another DNA tether, which is attached to a magnetic bead. And that magnetic bead, now we can just drop a magnet on here and we can pull on the protein. So we can actually play with this protein. I just find that amazing in and of itself. But in any case, we're doing that. And so now this protein is ensconced in a bilayer-like environment. It's a bicell. So in certain amphiphile detergent mixtures, you can get these bicells to form. It's little bilayer discs. So it's not a true membrane, but it's pretty close. We'll take it. Okay, so now we can pull on this protein and unfold it, and then when we release the force, it can't go anywhere. It's tethered, right? So it might as well just refold. You know, it doesn't have anything better to do with itself, and it certainly can't get into trouble with other membrane proteins. Okay, so that's the hope. All right. So the protein we have in there is a protein called GLP-G. I'm not going to tell you anything about it, other than that it's got 6 transmembrane helices, so it's a real membrane protein. And we're pulling on that. Now, I just want to point out that Daniel Muller has pioneered forced unfolding of membrane proteins using AFM. And what he does is he lowers an AFM tip onto the protein, and a miracle happens, and the protein sticks to the AFM tip, and then he can pull it out of the membrane. Okay? So he's done this a lot, and this is super cool. But it's not really a physiologically relevant type of unfolding, right? We want to see folding within a bilayer. That's second stage of folding. So that's why we've set this geometry up so that we're pulling along the plane of the bilayer. Okay, so this is what I expected to happen. So these transmembrane helices are fairly stable domains by themselves. And so I thought it wouldn't be too hard to pop a helix off and then maybe pop another helix off. And then maybe another one, and so you see fairly low force events, and it'd be it'd be non-cooperative. You see multiple low force events, it'd be fairly desultory unfolding. Okay, well, it's good to do experiments because that's not what we see. So, so this is just showing as we increase the force, the extension of that bead that we're monitoring here. Okay, so as we increase the force, you see increased extension, and this is just stretching the DNA. So we're stretching the DNA, stretching the DNA, stretching the DNA, and then, boom, that's where the protein unfolds. Okay, so we have to go to high force. We see this single unfolding event. So that's the protein popping. Now, so then we go to high force, and we're stretching the DNA again. Now, as we lower the force, what we'd love to see is for the protein to pop back. Then we could just monitor this equilibrium, and life would be good. That's not what we see. Basically, nothing happens. It doesn't refold. It doesn't refold. If we wait down here for a little while and pull again, the protein does refold. Okay, so because we can pull again and we see that the protein refolded. You see another pop. And we can do that like 40 times, so it's really robust. Okay, so the first disappointment is this hysteresis that we see. We wanted to see it pop back. It doesn't. Okay. Now, the other disappointment is that this is a very large extension. Okay, it's about 350 angstroms. Okay, so that is basically, this protein is stretched to a worm-like chain. So it's stretched so far that there's no way it's still in a bicell. So this is what actually happens. Okay, we pull on the protein harder and harder and harder, and then the protein explodes, all in one go, essentially. Let's do that again. Okay, all right, there we are. All right, so that's a disappointment because I want to study folding in a bicell, right? And this darn thing got pulled out of the bicell. Damn it. All right, so what are we going to do? We can get there. We can learn about unfolding by extrapolation, by unfolding in a bicell by extrapolation. Okay, so let's go back to this model, this sort of model of protein folding, this paradigm for protein folding, where we have a denaturation curve here. We add increasing denaturant, watch the protein unfold, jump to high denaturant, watch the protein unfold. We can jump back and watch the protein refold. Now, what we want to know is what are these, when we can learn rate constants, what we want to know is these rate constants in the absence of denaturant. We can learn that by extrapolation. So in simple cases, you can just plot these chevron plots. So this is just plotting folding rates here as a function of denaturant. And then these are unfolding rates as a function of denaturant. And to learn the rates in the absence of denaturant, you can simply extrapolate back to get the value in the absence of denaturant. And for the folding rate, you can do the same thing. Okay, so we can do a similar thing with, in our system, only here our denaturant is force. Okay, so that's what we've done here. So this is just the unfolding direction. As we increase the force, the probability of unfolding increases. In the refolding direction, as we lower the force, the probability of folding increases. Okay, and now in this case, so by extrapolation, we can learn the rates in the absence of force, but we also get another very important piece of information. So what we're doing here is when we add force, we're basically lowering the free energy difference between the native state and the transition state. Adding force, and the amount of amount of lowering is basically force times distance. Physics 101, right? Force times distance gives you energy. So we're lowering that energy by that amount. And so we can fit this to this Bell equation, and we get the distance to the transition state. Okay, so we get not only the rate in the absence of nature, but we also get the distance to the transition state. So in this case, it's about 15 angstroms. So in the unfolding direction. So from the folded state to the transition state for unfolding, it's about 15 angstroms. So that's a fairly small expansion. It's about the width of a transmembrane helix. Okay, so maybe you can imagine that's just popping a helix off and then the protein explodes. Okay, so let's think about sizes here. So the bicell is about 250 angstroms using the conditions that we used. The folded state of the protein is about 35 angstroms. So it's floating around in this big old bicell. When we get to the transition state, we've only expanded 15 angstroms. So that's well within the realm of, that can easily stay within a bicell. So we really think that going from the folded state to the transition state, we're still in a bicell. That's good. What about refolding? So when we go when we go to low force and watch the protein refold, the distance to the transition state is about 35 angstroms. Okay, so that would give us an unfolded state size of about 85 angstroms. Okay, that could fit well within a bicell. Okay, so at low force, we think that perhaps we are reforming the protein bicell complex. We can actually see that if we plot the extension of the protein as a function of force. At high force, it behaves like a worm-like chain, but as you drop the force, we see this transition at about 18 piconewtons. That's about a 100 angstrom drop. And so we think that and it also fits a model where we have helices. And so we think that basically is, at these lower forces then, the protein bicell complex is reforming. So we're refolding from a protein bicell complex. Okay, so let's put this together. So at zero force, the native state is more stable than the unfolded state. At high force, the unfolded state is more stable than the native state. That's why it unfolds. Okay, so let's fill in this picture. Well, we know what the native state looks like. That's just the crystal structure. The transition state, we learn about at high force anyway, is about 15 angstrom expansion. Let's imagine maybe we're popping off a helix. Okay? Once you get past the transition state, the protein explodes. Okay? So this whole protein bicell complex explodes. Okay? So from here to here, we're still in a bicell. After that, all bets are off. This part is not so interesting to me. If you're interested in this, you can read our paper because there's some stuff that happens here. It hiccups along the way sometimes, and we've characterized that. Okay, so along the way to the unfolded state, it hiccups a few times sometimes, and it's amusing, but not really of interest to me because I'm interested in folding in bicels. Okay, for the unfolded state, we think basically at low force, we reform this protein bicell complex, so we're seeing refolding from the bicell protein complex. Okay, so we can put these together, so we learn about the unfolding direction at high force and the refolding direction at low force, we think in bicels. We can put that together to build an energy surface. Okay, so from the ratio of the rate constants, we learn, so here's the native state, here's the native state, here's the unfolded state. From the ratio of the rate constants, we learn about the thermodynamic stability of the protein. That is, it's about only 4 kcals per mole, so it seems to be not a very thermodynamically stable protein. Nevertheless, this is high barrier to unfolding, okay? And that's why we have to go to these high forces in order for us to see the unfolding. Okay? And this barrier to unfolding is very close to the native state. And so that's going to tend to keep it in this native well. Okay, so this energy landscape that we see may have been worked on by evolution to prevent bad things from happening. So we're going to enter a realm of speculation here. So as this protein is being made, these transmembrane helices are being inserted. They get inserted one at a time. Now, we see this highly cooperative unfolding. Okay, suggesting that you don't have intermediates in folding, so it's sort of an all or none process. So that suggests that this protein has to wait until all the helices are made before the protein can fold into a stable state. And that may be important because if these helices start to assemble early, I don't know how to describe this exactly, but if these are a couple of helices, if they start to assemble early, oftentimes these helices, they're interdigitated, right? So a third helix has to come on. You would have to break that interaction in order for this other helix to get in there. So basically, you'd have to backtrack the folding. So if you form these stable interactions early, it could really slow the process of folding. Okay, so that's one possibility. Now, once this protein's in its native state, it's prevented from going back to the unfolded state or these partially unfolded states by this high barrier, this high kinetic barrier to unfolding. Okay, and this may be important then, of course, to prevent trouble with this unfolded state interacting with other proteins or even itself. Okay, so what we don't know at this point is whether this type of these features of this energy surface that we see for GLP-G are general for membrane proteins, or whether this is, just specific to GLP-G, because we haven't been able yet to look at many other membrane proteins. But I'm very hopeful that these kinds of techniques, these single molecule techniques that I described, will allow us to have a reasonable chance that we can pick a new membrane protein and actually learn something about it quantitatively. And so we are currently in the process of attacking these guys. We'll see what happens. This guy is got 15,000 atoms and it folds in 3 compartments of the cell. If we can do that, I think we can do just about anything. So we'll see what happens. We're going to give it a go. Okay, and with that, I will quit. And I just, again, thank the people that did this work that I talked about, and I mentioned them as I went through the talk. And thank you for your attention.